The rationale for this project is really to find new therapeutic strategies for our multiple myeloma patients, especially when they become refractory or resistant to the disease, when in clinic we don't have any other treatment options. So this research is based on the opportunity to find new therapeutic strategies for those patients that do not respond to chemotherapy anymore, and is really developing new compounds that can possibly be added to standard chemotherapy and resensitized to the treatment. In Moffitt, we have a very comprehensive multiple myeloma patient database that allowed us to identify an autophagy pathway that is overexpressed from patients while they become resistant to chemotherapy. And ULK3 is one of those proteins that increase its level of expression, going from the pre-malignant stage MGUS to later stage of the disease. So ULK3 is a novel protein that we associated to this pathway, and we think is a good biomarker for patients. So we were able to co-localize multiple myeloma cells in our patient tissue microarrays with this ULK3 protein, and also correlate this with the disease stage of the patient. And we took a genetic approach in the lab, ablating this protein from multiple myeloma cells, showing that actually those cells cannot survive without this protein. Therefore, in collaboration with our chemistry department collaborators, we designed novel inhibitors to basically inhibit the activity of this enzyme and hopefully kill multiple myeloma cells faster. So we took a preclinical approach, just developing this compound in our preclinical mouse model, where we see that those compounds actually are effective in reducing the tumor burden and increasing the overall survival of our animals. The strategy that we are using is really reporting this to our patients, and Moffitt is really benefiting a lot from this EMAS platform that is an ex vivo platform where we can test a combination of drugs and on patient samples. So we can actually test the efficacy of those drugs directly on the patient cells in their tumor microenvironment. So this is really the strategy that we are using that is novel and allow us to not test the drug directly into the patient, but actually pre-screen them in vitro. So we proved that this ULK3 protein is a good biomarker for multiple myeloma, and we are developing currently novel selective inhibitors for this enzyme, and we will continue to do so because there is really an unmet need and there is nothing out there on the market that could be used with this strategy. So our future direction is to continue studying why multiple myeloma cells become resistant to chemotherapy, to identify how this is happening through the autophagy metabolic pathway, and also to possibly identify a new compound that can be associated to standard of care chemotherapy in order to resensitize a patient to standard of care treatment.